So, good afternoon, students. Good afternoon, students.
Good afternoon, sir. Good afternoon, sir.
So, today again, we'll be having a very interesting class. I hope तुम्हें वो interest आ रहा है in monoclonal antibodies. शायद तुम्हें थोड़ी सी दिक्कत हुई थी वो समझने में the hat medium that uh, how the production of monoclonal antibodies. But I'm sure कल से जब तुम you are understanding the applications of monoclonal antibodies, uh, the interest is right back, right? Uh, Today again we are going to talk about other applications of monoclonal antibodies. So before I move ahead with more applications, I just want few of the students, if you can talk what applications we have discussed by this time. So just anyone, I just want few hands to be raised. अभी तक हमने क्या applications discuss की हैं monoclonal antibodies की? So students, this is very interesting topic, right? So तुम्हारी तरफ से भी that interest should be there. So what all applications have we discussed? Okay, Manaswini, I can see your hand. Shakti, okay. 2 students. In a class of 21 students or 22 students, just 2 hands being raised. So other 20. Okay, so Manaswini, you just talk about any 2 applications, and Shakti, you talk about any 2 applications. Okay, and when you are speaking, just switch on your camera. That would be good. Okay.
So
Yes,
can I start?
Manaswini, yes. Yes.
Yeah. So
Any 2 applications that you can
yeah.
think of?
So one application is that it's used in the Streptex kit to identify which strain of Streptococcus is infecting a particular individual. And then another application can be in immunopurification, where it's isolation of one particular protein from, let's say, any kind of sample, for example, a blood sample.
Okay, Manaswini, why purification? Why are we doing this purification? Any idea?
So my best guess is that once you isolate it, let's say you have to study the particular protein, then it's important to study the pure form of the protein to
Right.
understand its properties and everything.
Yes. So for a lot of research activities, you need to have a specific protein in your hand so that you can study it. Or suppose if it's a protein and you want to see its interaction with another protein or with another cell, then you need to have those purified proteins, right? So they can have a lot of applications. You can use it as a drug, you can be using it as a toxin, you can be for any purpose, right? So immunopurification becomes very important, and as I said in yesterday, that for research work, right? So you want to see if in a patient the expression of that protein has gone up or down, or the structure of that protein has changed, right? So what happens in some of the disease conditions? Like just for example, in a patient, the complement protein is not working, right? So you would like to see what has changed in that complement protein, right? So you can just take out that protein and then you can get it sequence. You can see what change have been there in that sequence or in the structure, right? So you can get a lot of information from that. So she talked about two things: streptex and uh, immunopurification. Okay, so Shakti,
Yes, sir. Am I visible?
yes, Shakti, you are visible,
Yes, sir. So it is also used in the testing of pregnancy, like in pregnancy kits. So
right?
it basically tests the hormone like human, I think it's HCG.
HCG, yes,
Yes. So there is an antigen and antibody interaction, and it is
okay,
also used in the testing of ABO blood groups.
right,
And also we talked about in the treatment of cancer, but also it is used for the detection of tumors. Like if we want to detect where the tumor is present in our body, so we can detect it.
right. So you also talked about so many applications, right? So you talked about therapeutics, you talked about ABO blood grouping, right? So and also pregnancy detection, right? So before I move ahead, I'll take an example and I'll show you a video of how the pregnancy detection kit works. Okay, and Manaswini talked about immunopurification. So one of the method that I discussed yesterday was using the magnet, right? So I'll show you videos on both these aspects, and then we will move ahead on other applications of monoclonal antibodies. So let me first show you how the pregnancy test works. Okay, so let me first show you the video on how the pregnancy test work. We have discussed it in theory, right? So we know that uh, what these kits are looking for. They're looking for the presence of human chorionic gonadotropin hormone, HCG. So this hormone is present in the urine when the female is pregnant. Okay. So let me share this video.
1350 BC in ancient Egypt. According to the Egyptians, all you have to do is urinate on wheat and barley seeds and wait. If either sprouts, congratulations, you're pregnant. And if wheat sprouts faster, it's a girl. But if barley, it's a boy. In 1963, a small study reproduced this test and found that it predicted pregnancy with a respectable 70% accuracy, though it couldn't reliably tell the sex of the baby. Scientists hypothesized that the test worked because pregnant women's urine contains more estrogen, which can promote seed growth. Now, it's easy to take this ancient method for granted because modern pregnancy tests give highly accurate results within minutes. So how do they work? Over-the-counter pregnancy tests are all designed to detect one thing, a hormone called HCG. HCG is produced in the earliest stages of pregnancy and starts a game of telephone that tells the body not to shed the inner lining of the uterus that month. As the pregnancy progresses, HCG supports the formation of the placenta, which transfers nutrients from mother to fetus. The test starts when urine is applied to the exposed end of the strip. As the fluid travels up the absorbent fibers, it will cross 3 separate zones, each with an important task. When the wave hits the first zone, the reaction zone, Y-shaped proteins called antibodies will grab onto any HCG. Attached to these antibodies is a handy enzyme with the ability to turn on dye molecules, which will be crucial later down the road. Then the urine picks up all the AB1 enzymes and carries them to the test zone, which is where the results show up. Secured to this zone are more Y-shaped antibodies that will also stick to HCG on one of its 5 binding sites. Scientists call this type of test a sandwich assay. If HCG is present, it gets sandwiched between the AB1 enzyme and AB2 and sticks to the test zone, allowing the attached dye activating enzyme to do its job and create a visible pattern. If there's no HCG, the wave of urine and enzymes just passes on by. Finally, there's one last stop to make, the control zone. As in any good experiment, this step confirms that the test is working properly. Whether the AB1 enzymes never saw HCG, or they're extras because zone 1 is overstocked with them, all the unbound AB1 enzymes picked up in zone 1 should end up here and activate more dye. So if no pattern appears, that indicates that the test was faulty. These tests are pretty reliable, but they're not fail-proof. For instance, false negatives can occur if concentrations of HCG aren't high enough for detection. After implantation, HCG levels double every 2 to 3 days, so it may just be too early to tell. And beverages can dilute the urine sample, which is why doctors recommend taking the test first thing in the morning. On the other hand, false positives can come from other sources of HCG, like IVF injections, ectopic pregnancies, or certain cancers such as uterine cancer or testicular cancer, making it possible for one of these tests to tell a man he's pregnant. The best way for a woman to find out for sure is at the doctor's office. The doctors are also looking for HCG, but with tests that are more sensitive and quantitative, which means they can determine the exact level of HCG in your blood. A few minutes can feel like forever when you're waiting on the results of a pregnancy test. But in that brief time, you're witnessing the power of the scientific method. That one little stick lets you ask a question, perform a controlled experiment, and then analyze the results to check your original hypothesis. And the best part is, you won't even have to wait until the next harvest.
So students, you just saw in this video, right? I hope now it's clear how it's working. So it's a full experiment that is happening on that strip, right? And there is also analysis. There is a control line, test line, and you are analyzing the result, right? So I hope this concept is clear, and this is just an example of a pregnancy test, but the same principle applies for any test. So even if you are looking for COVID, okay, so the principle remains the same, okay, uh, and this is how these are rapid, and I also said yesterday these are rapid diagnostic kits. Okay, so they give you the result in maybe 5 minutes or 2 minutes, right? But there are other methods like uh, um, ELISA. Okay, so in some of the cases, as mentioned in the video, if you need more accurate results, you can go to the doctor, and the doctor can prescribe a blood test, and you can also, like, because ELISA may be more sensitive than these diagnostic kits, right? So because every technique has its own advantage and disadvantage. Just for example, the advantage of rapid antigen detection is that it gives results within minutes, but it may be less sensitive. Okay, so if there is very less concentration of a particular protein or antigen, it may not pick it up. Okay, other technique like ELISA, it disadvantages, it will take more time, but advantages, it is more sensitive. Okay, so there are other methods like RIA, it is even more sensitive than ELISA. So every technique has its own advantage, disadvantage. Other advantage or disadvantage could be the price, right? So if you are going for RIA, it would be expensive, ELISA would be less expensive, and these rapid diagnostic kits are like just 100 rupees or 70 rupees, like that, right? Okay, so that was one thing. Another, we talked about immunopurification using magnetic beads. So let me just also show you um, this process. So you can see with the help of this magnet, right, all the cells which were tagged using monoclonal antibody, they have come at one place. Okay, and then you can remove rest of the solution. Okay, and then whatever will be left in this solution are these cells. Okay, so rest of the fluid has been sucked up. So you are just removed. You are just left with these cells which you actually required. Okay, and as soon as you remove the magnet, you can have the cells back in the suspension, and that you can use for your purpose. Okay, so students, I hope both these applications are clear to you. So Manaswini talked about this immunopurification, and I have shown you how we can do this using magnetic separation. Okay, and uh, Shakti talked about this pregnancy detection. So again, I have shown you the video of that. So I hope both these applications are clear to you right now, right? Any questions on this?
Sir, can you share the links to both the videos so that we
Yes,
can use it for future? Yeah.
yes, I have put the links in the slides itself. So when you'll get the slides, you'll get the link as well. Okay, I'll just show you that I have put it in the slides. Okay,
Okay, okay. Thank you, sir.
yeah. Okay, so you can see I have put this link here. This is for pregnancy detection, how it works. Okay, and the magnetic separation I have put here. Okay, and we'll talk about this flow cytometer in detail, maybe at the end of this semester, because this is in the fourth module. So we'll talk about this later in details, right? But at least now you know that monoclonal antibodies are required in flow cytometry for tagging and separation of protein of your interest. Okay, how it works is what we will be discussing later. Okay, now other applications uh, are like monoclonal antibodies are used to detect the presence of particular proteins. Okay, so one of the tests is Western blot, and I'm sure you have read Western blot. कहीं ना कहीं किसी उसमें you may have read Western blot, right? Have you read Western blot?
Yes, sir.
Right? So you know what is the purpose of Western blot? So why do we
The
carry out?
detection of a protein.
Yes, so Western blot is basically used for detection of proteins, right? And so students, what happened? Like if suppose I give you a cell, okay, and if I ask you to find a particular protein, then what happens, right? You will take that cell, you will prepare that cell, and then ultimately you will run it on the SDS page. right? So I'm not going to discuss this SDS page. I'm sure you may have read SDS page, right? So SDS page is usually used for separation of proteins. Now the problem will be, in a cell there may be so many different um, proteins. So what will happen on this gel? You will see lot of different bands, right? So there will be lot of bands, right? Because one cell will obviously have a lot of different bands, but you want to see whether, for example, this COX-1 protein is present or not. Okay, or for example you want to know if beta actin is present or not, right? So what we do is we transfer it onto the membrane, and then we incubate it with an antibody, monoclonal antibody, and if that protein is present, that protein will bind, okay, with the monoclonal antibody. And from hundreds of proteins you will find whether your protein of interest is present in that sample or not. Okay, so Western blot and immuno.blot test, they detect the presence of protein on a membrane. Okay, so this is how we carry out Western blotting. I'm not going to discuss how Western blotting is done, or what is there in the SDS, how we prepare the sample. But I'm sure you know it, you have read it, and you know the details of it. Okay, then monoclonal antibodies are also useful in immunohistochemistry, okay, which detect antigen in fixed tissue sections. Okay, so now just for example, when we take biopsy sample, right, we take some tissue sample, and you want to see the presence of specific protein, okay, then you can do histochemistry, right? Just for example, a cancerous cell, if you are looking for the presence of cancerous cell in a tissue, right, so you can target the cancerous cell, and then you can visualize. So you can see this picture over here, right? So you can see these green dots, right? So this is a protein which you can see is present inside the cell as well as on the outer side of the cell, right? So it helps you uh, to know the uh, presence and its location. And these red proteins, they are just present inside the cell. Okay, so you can get to know the specific protein present in a fixed tissue section. So if you are using fixed tissue sections, then it is immunohistochemistry. Okay. Then immunofluorescence test, which detect the substance in a frozen tissue section or in a live cell. Okay. So if you want to see it in uh, a live cell, okay, then you can use the technique of immunofluorescence. So again, I'm not discussing these techniques in detail, I just want to discuss the application part, right? So basically you can use monoclonal antibody to find your protein of interest in either the fixed tissues or even in live cells. Okay, so if it's a live cell, then the technique is immunofluorescence test. If it's a fixed tissue section, it is immunohistochemistry. If you are trying to find it on a membrane, then it is Western blot. Okay, so students, I'm sure you have not read many research papers, but if you will read research papers, lot of research papers you will see this kind of image, right? And in lot of papers you will also see this kind of image, right? So it shows the presence of that particular protein or whatever we are looking for. Okay. Okay, so these are other applications of monoclonal antibody another very important application that has come up is in imaging. Okay, so now we are using radio labeled monoclonal antibodies. Okay, so students, the FC region, okay, you can tag some radio labeled compound to the antibodies in the FC region. Okay, and then you can use in vivo for detecting or locating tumor antigens. Okay, now this permits early diagnosis of some tumors. So uh, the tumor antigens they shed late, right? So by the time we come to know about cancer, it has already reached the third or the fourth stage, right? I'm sure you have read the news of Sanjay Dutt having cancer. So he came to know when it was very late, right? So a lot of time we get to know about cancer at a very late stage, okay, because those tumor antigens are shed in the blood very late, right? So if we use radio labeled monoclonal antibodies, right, so this will permit us early diagnosis of some tumors. Now, just an example is monoclonal antibodies to breast cancer cell is labeled with iodine 131, right, and injected into blood to detect spread of tumor. So, sometime what happens, the tumor, then it becomes metastasis. So, you never get to know where those tumor cells have moved into your body. Okay. So, monoclonal imaging technique can detect breast cancer metastasis that would be undetected by other less sensitive scanning techniques. Okay. And students, you can see a picture here, right? So this is a picture of a mice, okay, and it was given monoclonal antibody labeled, okay. So it shows you the area where it is having the cancer, okay. So the tumor is here, and you can see the presence of this tumor, right? And then this mice were given the anti-cancer drugs, okay, and then you can see over a period of time it is reducing in size. The tumor is reducing in size, right? So we can uh, check uh, the presence of tumor, okay, and we can also target these tumors. We can add some drug to monoclonal antibodies to kill these tumors, okay. So is it clear, this one, how monoclonal antibodies can be used in imaging and detection of cancer? Any questions on this? No questions? Okay, so let's see other applications, and this is of immunotoxins. I'll just take attendance break. You can just revise what I have discussed, and if you have any question, you can ask. Uh, okay, Yuvraj has put a question, sir, why other scanning techniques cannot detect the breast cancer? Okay, so these techniques they can, but usually not in the initial stages. Okay, as I said, in the initial stages, it is difficult to locate these tumors. Okay, and you know what happens initially? You don't have any sign or symptom. Okay, so we actually don't go and visit the doctor. That is one issue, and even if we try, then most of the techniques will not uh, detect. So one of the things, as I said, uh, that usually we go for the blood test, okay, uh, in which the antigens are shed by the cancer cell, okay. So you cannot actually detect them because there can be a lot of issues, like they are in very less number, okay. So the techniques are not sensitive enough to pick them, okay, and sometimes they are not in the initial stages shed, okay. So in that case you will miss these tumors in the early stage, right? So the question is, question should be, why other techniques cannot detect the breast cancer at an early stage? Okay, later stage, yes, it can be detected, but not in the early stage. Okay. Okay. So let's have attendance, and you all can think. If you have any question, you can ask. And let me start with attendance. So Adish,
Present, sir.
okay, just wait. Okay, Atulba,
Present, sir.
Ayushi,
Present, sir.
Sam,
Present, sir.
Sam, you are not visible.
So am I visible now?
Yes, Sam, you are visible. Okay. Devanshi,
Present, sir.
Ishita,
Present, sir.
Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita,
Present, sir.
Nandini, Nandini, Nehal,
Present, sir.
Rupal,
Present.
Sahiba,
Present, sir.
Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir.
Vivek,
Present, sir.
Yukta,
Present, sir.
Yuvraj.
Present, sir.
Okay. Okay, so Yukta, your connection is fine now? Okay, Nandini, I got your message. Okay, so students, let's come back to the application part of monoclonal antibody, and we are going to talk about another very important upcoming field for the use of monoclonal antibodies for therapeutics. Okay, and let me show you this. So monoclonal antibodies can be used as immunotoxins. Okay, now this is very interesting. How we are using a toxin and how we are using antibody, making a complex of them and using it for our benefit. Okay, so immunotoxins are basically tumor specific antibody. Okay, tumor specific antibody means they will go and bind to the cancer cell. Okay, and in immunotoxin we are also adding a lethal toxin. Okay, now why we are adding these two things? So tumor specific antibody, what will it do? It will go and bind to the cancer cell. Okay, the advantage, it is only going to target the cancer cell and it will not affect any other cell, right? So I'm sure you know कि जब हम cancer treatment करते हैं, right? So we either use the radiotherapy, right? Or we use the chemotherapy. And in both the cases, even the normal cells are affected and they have more side effects, right? But if you use tumor specific antibody, they will only target the tumor cells and they will not go and bind to any other cell, right? So this is the purpose of having tumor specific antibody, and to this antibody we are binding a lethal toxin. Okay, now what will this toxin do? It will go and kill the cell. Okay, which cell? The tumor cell, because the antibody is bound to that tumor cell. Okay, so this combination of tumor specific antibody plus lethal toxin, it is used as a therapeutic reagent. And let me show you some examples of toxins. So you have, like for example, uh, the ricin, okay, or the Shigella toxin or the diphtheria toxin. Okay, now these toxins, they can inhibit protein synthesis. Okay, so these are toxins, they can inhibit protein synthesis. So we can use these toxins and then target the protein synthesis mechanism of the cancer cell. Okay, तो जब कैंसर सेल में प्रोटीन सिंथेसिस नहीं होगा, so the cell will die. Okay, so just see how we are using it, but before that let's see the structure of the toxins. Now this is your ricin toxin, this is your Shigella toxin, okay, this is your diphtheria toxin. Just see them. So they have two chains. One is this pink chain, another is this yellow chain, right? And all these have this. So diphtheria one, okay, so both yellow and the pink chain, right? And if we talk about immunotoxin, what we have done is we have taken this tumor specific antibody, and to this, to the FC region, we have added the toxin chain of this toxin, right? And now this can be used for the treatment of cancer. Okay, now let's just understand this again. So these toxins which I just showed you the pictures, I said there are two chains. One is the yellow one and the pink one, right? So these toxins have two functionally distinct. One is the inhibitory chain or the toxin, and another one is the binding chain. Okay, so you can see, if I take example of diphtheria toxin, it has this toxin, okay, and this is the binding chain. Now the role of the binding chain is to take the toxin and to bind the toxin at its receptor. Okay, binding chain का काम क्या है? It will go and bind to the receptor on the target cell. Okay, and what we are doing in immunotoxin, we are removing this binding chain, okay, and replacing it with a monoclonal antibody which will target a specific epitope. Okay, so now immunotoxins, they are prepared by replacing the binding chain with monoclonal antibody that is specific for the tumor cell. Okay, clear? Now this attached monoclonal antibody will deliver toxin chain specifically to tumor cells and cause its death by inhibiting protein synthesis. Okay, and how this happens I can show you with the help of this picture. So just focus on this side. Okay, so this is your diphtheria toxin. As I said, the binding chain will help it in binding to the cell through its receptor. Okay, once this binding occurs, this complex, this toxin is taken inside by endocytosis, okay, into the endosome, okay, where the toxin is released into the cytosol. Okay, now your toxin from outside, it has gone inside and it is released into the cytosol, uh, where it inactivates the elongation factor 2. Okay, so I really don't know if you have read the protein synthesis. Okay, so there are elongation factors, right? So what this toxin does is it inactivates the elongation factor. Okay, and because of which the protein is not synthesized. Okay, it blocks the protein synthesis and the cell dies. Okay, now if we use immunotoxin, it will bind to only tumor specific antigens, right? So this is specific, it will go and bind to the cancer cells. Same thing will happen, it will be endocytized, it will get into the endosomes where there will be presence of various enzymes that will cleave this disulfide bond. This toxin will then be released into the cytosol. It will then inactivate the elongation factor 2, and therefore there will be no production of proteins and the cancer cell will die. Okay, so this is how we can use monoclonal antibodies for the treatment of cancer, right? Is the concept clear?
Yes, sir.
Any questions on this? Is it clear?
Yes, sir.
Yes, sir.
Okay. Okay, then I can show you some examples of monoclonal antibodies which are in use or which are under clinical trials. Okay, so So these monoclonal antibodies, apart from killing the cancer cell, they have been used for various purposes, like to suppress the immune system, right? So there are a lot of times when uh, we need to suppress the immune system, okay? And to suppress the immune system, we can use these monoclonal antibodies. I'll just talk about few examples. You don't need to get into the details of the mechanism of different monoclonal antibodies, okay? But just for your understanding, just to make you understand that it can be used in different ways, I'm showing you these examples, okay? At your level, you don't need to get into details of their mechanism, okay? But yes, when you are in masters, we will get into more details of these pathways, okay? So uh, we can talk about this muromonab, okay, which is also called OKT3, right? It binds to CD3 molecules on the surface of T cells, okay? So this monoclonal antibody will specifically target the T cells, okay? And this is used to prevent acute rejection of organs. So, right? So suppose if a person has had organ transplant, then we give OKT3, okay, and this will bind to T cells, uh, and this will prevent acute rejection of the organ, okay. The humanized version shows promises in inhibiting autoimmune destruction of beta cells of diabetes. So it is also helpful in diabetes, type 1 diabetes. Okay. Another example is infliximab, okay, or this Humira. Okay. It is very popular. It binds to tumor necrosis factor alpha. So it binds to TNF alpha. Again, you don't need to know much about TNF alpha, how it works, what it does, but you can just remember that it binds to TNF alpha, and it is being used for treatment of rheumatoid arthritis. Okay, then you have omalizumab. Okay, it binds to IgE. So you know what IgE causes? It causes hypersensitivity type 1 reaction. We have already discussed that, right? So what happens when that IgE, it goes and bind to the mast cells? You remember we have already discussed this in detail. When IgE, it goes and binds to the Fc epsilon R on the mast cells or the basophils, then the mediators are released from these cells which cause type 1 hypersensitivity reactions, right? So if you put this monoclonal antibody, it will go and bind to whatever IgE is present in your system, okay, and it will not let uh, degranulation occur. Okay, so this monoclonal antibody can be used to prevent hypersensitivity type 1 reactions. It can also be used in case of allergic asthma. Okay, so because it is allergic, then obviously there is the role of IgE. So whenever there is allergic asthma, this monoclonal antibody can be used. Okay. So similarly, there are many other examples. One of the very important monoclonal antibody in use is rituximab. Okay, now this binds to CD20 molecule on B cells and is used for the treatment of B cell cancer. Okay, uh, so I don't know if I need to discuss all these. This is fine. I think uh, uh, it's fine. Okay, so you don't need to get into all these uh, monoclonal antibodies because it's a very long list. I'll show you that list, but you don't need to remember every type. Okay. Now there are some angiogenesis inhibitor. Again, I don't know if you know what is angiogenesis. It's basically formation of new blood vessels in case of cancer. So you can target vascular integrins, right? Um, so these are used for treatment of tumors. Okay. So there are a lot of examples which I'm not going to discuss at your level. So I'll just stop here, and in the next class we'll talk about the different types of monoclonal antibodies. Okay. So students, if you have any questions, you can ask, or else I'll end the class now. Anything you want to know?
No, sir.
Okay, so I'm ending the class now. Okay, see you in the next class.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Okay,
Thank you.
students. Okay.